We are at 11 o'clock. Good to see y'all. We have lecture today and of course laboratory today, but Friday is Cesar Chavez Day, so no, the school is closed on Friday. And then next week is spring break. So obviously no school next week, and then we'll come back on Monday and kind of take up where we left off on this class. All right, well, let's see. We're on mitosis, which, remember, is the type of nuclear division that cells do, where the daughter cells end up with the same number of chromosomes as the mother cell. And let me do a quick recap on the chalkboard here. Before a cell undergoes mitosis, and remember again, mitosis is the nucleus dividing. It's a type of nuclear division. Before a cell undergoes mitosis, the chromosomes have to duplicate, meaning that each chromosome, instead of being just one centromere with its one chromatid of double-stranded DNA, has to turn itself into a duplicated chromosome, where you get one centromere but connected to two chromatids, two pieces of double-stranded DNA. And we call these sister chromatids of the duplicated chromosome. How many chromosomes here? One, right, because remember, don't be fooled by the two chromatids. If I ask you to say how many chromosomes, just count the number of centromeres, so it still counts as one chromosome. Let's see, the way this works is like this, that little bubbles of the double-stranded DNA come apart from each other all along the chromatid. Those are called origins of replication, or sometimes replication bubbles. You might get something like that. And actually, I'm only showing a couple of them here, but in reality, there might be hundreds of these replication bubbles all along the chromatid. All right, and in these replication bubbles, the part right here where the double-stranded DNA is moving apart and turning into single-stranded DNA, those are called replication forks. So each bubble has a fork that on its left-hand side and a fork on its right-hand side, like there and there, the replication forks. And what's happening there is like I said, the double-stranded DNA is moving apart to become single-stranded DNA. It's just like a zipper. So I put my zipper down, the double-stranded part right there goes into two single-stranded parts, like that. And the reason why it does that is there's a team of enzymes that are responsible for making the complementary strands to each one of these that eventually will give us the two chromatids from the original one chromatid. And the major enzyme involved is called DNA polymerase 3. That's the enzyme that actually rides along the single-stranded parts and makes the complementary strand. There's other enzymes involved. There's primase and ligase and another DNA polymerase. Actually, it's called DNA polymerase 1 that helps get rid of the primers. But just focusing on DNA polymerase 3, one of the DNA polymerase 3 enzymes makes the new complementary strand in the direction of the replication fork, and the other DNA polymerase enzyme that's on this template strand has to move away from the replication fork. So it actually makes the new complementary strand in short discontinuous segments called Okazaki fragments. And the reason why you have that is the DNA polymerase 3 enzymes have to construct the new strand in the 5' to 3' direction. And for this template strand, that's in the direction of the replication fork. But for this template strand, that's away from the replication fork. And so you get this discontinuous making of the new complementary strand on the lagging strand, which is the bottom one there. And those Okazaki fragments, which are the light blue-colored ones on the bottom strand, on the lagging strand, will eventually be joined together into one continuous one continuous piece of DNA strand after those primers are stripped off and replaced with DNA and ligases has joined them all together. All right. Sound all right? So when all is said and done, well, actually, you can see it here on the computer screen. Let's see. So there's the original unduplicated chromosome with its centromere. There are those origins of replication opening up, and then the DNA polymerase III working into replication forks eventually starts filling in the complementary strands. And as more and more DNA gets opened up at the replication forks, the origins of replication bubbles get larger and larger until they eventually meet each other. And eventually you get a duplicated chromosome out of the whole process like that. And remember this term, semi-conservative replication. What they mean by that is that each chromatid has one of the original old DNA strands and one newly synthesized DNA strand. Alrighty, let's see. Yeah, so there's the duplicated chromosome that you end up with. Maybe I never really explained, well, why does the cell do this before undergoing mitosis nuclear division? Well, this, actually, before I get into that, I was going to tell you about mutations. Sorry, I got a little out of order there. Here's the simplified chromosome duplicating. The two parts come apart like that. There's the DNA polymerase enzymes coming in. Let's fast forward slightly on this part. There we go. So the job of these DNA polymerase III enzymes is to make the complementary strand. So they ride along their template strands, just laying down the complementary nucleotides. Every now and then they'll make a mistake while they're doing it. And I think I should show you right here. There's the mistake right there. The original DNA strand had a C there, so there should have been a G there, but it just made an error when it did that. DNA polymerase III is actually fairly good about not making errors. I think last time I mentioned that it had an error rate of only 1 in 10 billion nucleotides. And actually, after I said that, one of your students here came up to me afterwards and said, I heard a slightly different number than that. And so I looked it up a little bit on the internet. I think my 1 in 10 billion was incorrect. It's closer to 1 in 10 million, the error rate. But anyway, the point is that DNA polymerase III has a relatively low error rate, but it does have an error rate, so occasionally it does end up changing the sequence of the gene. And that is called a mutation. Any change in the DNA sequence of a gene is called a mutation. And they don't happen too much, but they do occur. DNA polymerase III sometimes generates some mutations when it's replicating the DNA. There's radiation in the environment. There's cosmic rays, for instance, coming down from outer space that constantly hit us. The dirt has a small amount of radioactivity in it. We get exposed to X-rays and ultraviolet radiation from the sunlight. Certain chemicals in the foods that we eat or might otherwise get exposed to can cause mutations also. So not often, but they certainly do occur. And they can change the amino acid sequence of the proteins that come from the gene, because of course the sequence of the amino acids is based on the sequence of nucleotides in the gene. So, the gene up on top of this figure was not the one that had the mutation. So the gene up on top would make the normal functional enzyme that it was supposed to make. But since the one on the bottom had a mutation, it would not make the same protein as the gene up on top. Or to use a term that we introduced earlier, a new allele has been created. Alleles are slightly different versions of a gene. So what's the effect on the protein going to be? Well, almost always it's going to be harmful to the protein's function. Why? Because mutations are random. There's nothing selecting which nucleotide is going to get changed. And so it's a completely random change. And for anything that has lots of regions or lots of parts that have to work together, like these have to work together to form the binding site, if something's complex and has lots of different parts, if you make a random change, you're probably going to wreck its function. To make an analogy, if I open up my computer here, which is also a complex device, and randomly grabbed one wire, gesundheit, and just kind of randomly stuck it into another part of the computer, I'm probably going to burn the computer chips out. It's not going to improve my computer. Or if I open up my car, the engine, and randomly grabbed a hose from this side and stuck it in some other part of the engine, I'm probably not going to improve my car's performance. And so the vast majority of the time, mutation is going to decrease the protein's functions. In some cases, just render it completely unfolded, and so utterly take away its function. But that's not always the case. Very, very rarely a mutation will improve a protein's function. It might, for instance, change an amino acid so that the protein has a better binding site, a better active site than it did before, or it might make the protein fold in such a way so that a new binding site forms that can carry out some different chemical reaction. A new enzyme might be formed. I guess you could make an analogy again with your engine. If you reach in your engine and you randomly change something inside your engine, you're probably not going to do your engine any good. But very, very rarely you might, I don't know, improve the timing of the spark plugs or something like that, or in some way improve your gas mileage. It doesn't happen often, but it could happen. When very rarely these good mutations do occur, that's actually important for the evolution of the species, because the idea is this, that when an organism has one of these bad mutations that in some way harms the organism, its metabolic reactions don't work as well as they're supposed to, and so those ones tend to die off and not produce very many offspring. But if it's a good mutation, then that gives the organism that has that mutation some sort of advantage, and so it's able to survive better and make more offspring. And since we're talking about genes, which gets passed on to the offspring, that successful organism with its good mutation would pass that mutated gene on to its offspring, which would also be more successful because they have the good gene, so they pass it on to their offspring, and pretty soon the entire population of whatever species we're talking about will eventually, over a few generations, inherit that good gene because it helps them survive, and then the species is improved and changed. This is supposed to be the new improved protein from that mutation right there. And so over very long periods of time, over hundreds of thousands or millions of years, species do evolve slowly as they slowly accumulate mutations that benefit that particular species in that particular environment. And that in essence is the theory of how the theory of evolution works.
Is there any way for an organism to detect mutation?
To
detect like a mutation? It just happens, right?
Yeah, I mean, can the organism itself somehow note that it has been mutated? It can, yeah. Matter of fact, let me click back on something here. Okay, so notice that I said that here's a mutation right here. The cell would be able to detect that the nucleotide here is not matching up with the nucleotide there, because the cell does know that C's and G's are supposed to be opposite each other, and A's and T's are supposed to be opposite each other. But the cell doesn't know whether the A is the problem that's supposed to be a G, or whether the C is the problem that's supposed to be a T. All it knows is that they're not matching up. And so the cell essentially makes a random decision and will correct it. And half the time it would wipe out the A and replace it with a G, but the other half of the time it will wipe out the C and replace it with a T. And so I guess in a sense it can detect mismatches in the two bases, but it doesn't know which one's the correct one, which one's the non-correct one. Yeah, that's a great question. Okay, so mutation almost always bad, but when the good ones come along, those are the ones that allow a species to evolve. All right, getting back to mitosis for a moment. So this is supposed to be a cell, and Remember what I said a few minutes ago, that the chromosomes duplicate before the nucleus divides. This is sort of a precursor step to before the nucleus divides. And so let's watch that taking place. The cell wants to go through mitosis, so it duplicates its chromosomes. Now here I'm showing the chromosomes. I didn't show any replication bubbles when those chromosomes duplicated, but that's what the point is. Eventually the chromosomes duplicate. I hate to sound too repetitive, but how many chromosomes in this cell? 2, right? Don't be fooled by the chromatids, still 2 chromosomes. Okay, now that the cell has duplicated its chromosomes, now it's ready for the first step of division, which is nuclear division. And so watch what happens. The centromere split in nuclear division and move towards opposite poles of the cell. Now how many chromosomes? 4, right? Because when counting chromosomes, count the number of centromeres. Let me run that back again. All right, so there are our 2 duplicated chromosomes, but when we say the cell undergoes nuclear division, what's actually happening is the centromeres are split.